Hello, everyone. Welcome to this keynote session. We're going to be hearing from Tschida, who has just started a new lab at Cornell University in the Department of Psychology. And she's going to be telling us about vocalizations in mice and how we can learn about communication through them. So, Katie, please go ahead and take it away.
Okay, great. Let me get my screen shared with you all. Okay, make this larger. Okay, great. First of all, thanks a lot for the opportunity to tell you all about my work. Yeah, my only regret is that I can't see you guys in person. I'm never going to get over the weirdness of doing talks on Zoom. But I'm nonetheless very excited to have a chance to tell you about what I've worked on and what I'm going to be working on now. And so what I'll tell you about today, it's really a few things. So I'm going to tell you first about a story that's complete, but although many have heard it before, it does set the stage for everything that follows. So I'll do a brief recap of our story where we went into the midbrain and characterized cells within the midbrain of the mouse that are really essential for the production of ultrasonic vocalizations. Second, I'll walk you through a story that is largely complete, but still not 100% complete. And then finally, I'll end with some current directions that my lab is exploring at Cornell. And before jumping into the data, I'm actually going to tell you a little bit about kind of what I've done before and how I arrived at the line of inquiry in which I'm involved right now. So I have a really long-standing interest in social behavior and in vocal communication in particular. And I think it goes without saying that vocalizations are really important throughout the animal kingdom for survival and reproduction. In a small number of species, including humans, of course, we learn our vocalizations. And I actually got my start in my PhD work. I worked with zebra finches and other species that learn their vocalizations. And I studied the neural circuits important for singing and song learning in the zebra finch. And, kind of over the course of doing that work, I became really interested in the neural circuits that are important for the production of innate vocalizations. So of course, even in species such as humans and songbirds that produce learned vocalizations, we also produce innate vocalizations. So, in our case, it's things like laughing, crying, and I would expand from there, to even the emotional content of our learned vocalizations. And so, while there are a few species that learn vocalizations, the vast majority of species produce innate vocalizations that communicate emotions, internal states, affective states, etc. And I became really intrigued to understand the neural basis for these vocalizations because I don't think it's a crazy idea that the circuits would be really evolutionarily more ancient and highly conserved across mammals and probably across vertebrates as well. So the stories I'll tell you about today involve my work studying the neural circuits for vocalization in the mouse. There are a couple of really great reasons to study these questions in the mouse. One, of course, is that we have this massive arsenal of genetic tools at our disposal when we work with the mouse. And the second is the behavior itself is really great. They produce a really rich repertoire of vocalizations in a variety of social contexts. And I've listed some of them here, but this certainly is not an exclusive list of the contexts in which mice vocalize. But they do vocalize during courtship. They vocalize during same-sex social interactions. And then a final example is that mouse pups vocalize, and they do this particularly when they're isolated from the nest. And in the case of pup vocalizations, it's actually occurring when they become cold, they can't thermoregulate, and they engage in this laryngeal breaking behavior that essentially pushes out a USV with exhalation. So I'm going to play a video for those who maybe haven't seen or heard a mouse USV. And so let me just say before I start the video, when we tested it, the audio was a bit loud. So please you can turn down your volume just a bit, just to be safe. And And so what you'll see is a male mouse interacting with a female. Don't worry, it will be very obvious who is who. And then scrolling across the bottom, what you're going to see is a spectrogram, so a plot of time on the x-axis and frequency on the y-axis. And so the vocalizations will be streaming across in this spectrogram. And then there's also some audio. And so that's been pitch shifted so that we can actually hear the vocalizations. And so what I want you to appreciate is they kind of sound a bit like a songbird, and that they accompany a lot of other non-vocal social behaviors. So let's give this a whirl. Okay, so I hope you all could see the male is very vigorously courting the female. And, you know, a couple of other features to note are that these ultrasonic vocalizations, they are comprised of individual syllables that are produced in long strings. So there are these long vocal bouts that are comprised of individual elements. And there's really a lot of acoustic complexity and variability from element to element. Okay, so now that you've seen the mouse vocal behavior, we can talk a little bit about the neural circuits. Let me be the first to say, we were not the first people to ask this question, what are the neural circuits for innate vocalizations? And in fact, there's a really rich and extensive literature dating back to at least the 60s and 70s, implicating a part of the brain, a part of the midbrain known as the periaqueductal gray, which we'll abbreviate as the PAG throughout the talk, in the control of innate vocalizations. And so in a variety of species, really every one that has been tested to date, to my knowledge, lesions of the PAG abolish the production of innate species typical vocalizations, whereas activation of this structure is capable of eliciting the production of species typical vocalizations. And then another feature to note is that, of course, activation of other parts of the brain, and in particular parts of the forebrain, is capable of eliciting vocalization. But activating these regions no longer works. It no longer elicits a vocalization if the PAG isn't intact. So the thing to remember is that, from all of this previous work, we have a really good reason to believe that the PAG is an important hub in the circuits for the control of vocalization. Okay. And so, we've known this for a long time. Nonetheless, the PAG is a heterogeneous brain region, and we haven't made much headway in identifying and characterizing the specific cells within the PAG that are important for the control of vocalization. And that's really a critical thing to do to really get specifically, outside of the PAG, who provides input to those cells and who do those cells talk to better understand the nitty-gritty of how these circuits work. So the goals of this first study that I'll tell you about, which was published last year, are threefold. One, we wanted to ask whether we could identify neurons within the PAG of the mouse that are important for the production of USVs. And so we'll call these neurons PAG USV neurons. If we can identify these cells, of course we could, or I wouldn't be telling you anything. We wanted to ask whether they were specialized for vocalization, or alternatively, whether they controlled a variety of social behaviors. And then finally, we wanted to understand how they contribute to vocalization. So what I mean by that is, do these cells gate the production of vocalization? So potentially their activity is sufficient or necessary for vocalization. Or alternatively, are they important for the fine temporal patterning of individual vocal elements within a bout of vocalization? So those are the questions we'll walk through. Okay, so as I said, the PAG is functionally heterogeneous, and this video is really designed to illustrate that to you. So, a lot of, there's been a huge proliferation of studies in the last 5 years, especially implicating the PAG in a variety of behaviors. It's kind of to the point, like, pick a behavior, you can find a study involving the PAG and that behavior. So of course, vocalization, hunting, fear-related behaviors such as freezing and fleeing, pain processing, autonomic responses, you name it. Okay, so what happens then if, without regard to the functional identity of cell types, if we just plunk down into the PAG and if we activate cells using optogenetics. So here I'm showing you we've expressed channel rhodopsin non-selectively within the PAG, and we're going to activate this part of the brain with light. What do you get? So same warning as before, the video might be a little bit loud. Okay, so we get something very dramatic. Certainly this mouse does produce ultrasonic vocalizations, but you can see we're not getting just that behavior. And so in this particular example, the mouse engages in really ballistic locomotion, escape-like movements, right? So we're getting a mix of behaviors. And in fact, if you activate the PAG optogenetically with chemogenetics, et cetera, you always get this mix of behaviors. And so that, of course, wasn't our goal. What we really wanted to do was to really get selectively at those cells that are important for vocalization. And so to do this, I took advantage of a method that had been developed. It was developed in my postdoctoral lab and the lab of Dr. Fan Wong, right around the time that I was beginning my postdoc with her. So my good fortune. And the method is called CANE. The important thing to know is that it allows us to express genes in neurons that were recently active during the behavior. And I'll run you through the details. So this strategy depends on a transgenic mouse and also on a designer virus. So the transgenic mouse is called the FOS-TVA mouse. The entire strategy relies on using FOS, which is an immediate early gene, as a proxy for neural activity. So in this transgenic mouse, when the FOS promoter turns on, in addition to driving expression of FOS, it also drives expression of an exogenous receptor, which is known as the TVA receptor. And that receptor is destabilized, so it's not permanently expressed, but it's going to turn on with FOS and then come back down again after FOS turns off. So then in this transgenic animal, it engages in behavior, there's neural activity, and active neurons are going to transiently express this TVA receptor. We then go in with a designer virus that can selectively infect neurons that express this exogenous receptor. And so that's how we're going to selectively express genes in these neurons that were recently active during the behavior. And so by using viruses, we can drive the expression of any number of genes in these neurons, fluorescent proteins, optogenetic tools, et cetera. So that's the overall strategy that we took. And so in the first experiment I'll show you, this is going to contrast with the video that we just watched. What we did was we used the CANE strategy and we expressed channel rhodopsin, this time selectively in PAG neurons that were active during the production of USVs in the mouse. Okay, so in this case, we did get vocalizations, but we did not get all the other accompanying fear-related behaviors. So from the behavior, it does appear that we've really selectively isolated the cells important for vocalization. I don't have time today to show you the full characterization that we did here, but I wanted to show you a quick comparison of the spectrograms for one animal of, on the left, USVs that were elicited by optogenetic activation of these PAG USV neurons, and on the right, female-directed USVs that were produced naturally by that same animal. And the upshot of this illustration, and also our more in-depth analysis, was that the optogenetically elicited USVs were quite natural, as opposed to looking like very artificial and acoustically abnormal. So then we have established that activation of these neurons is capable of eliciting USV production. What about silencing? Are we able to block the production of USVs? And so in this experiment, we used, again, the CANE strategy. This time, we expressed tetanus toxin selectively in PAG USV neurons. And so this is going to effectively silence those cells by preventing them from releasing neurotransmitter. And so what the experiment that I'm showing you here is we've taken male mice. We measure the number of USVs they produce toward a female prior to the viral injection. We wait 2 weeks, and then we do a second measurement post-viral injection. And in this plot on the left here, you can see that in the vast majority of animals that we ran through this experiment, their USVs actually are completely abolished. They go all the way down to 0 or very close to 0. So silencing these cells has a very, very dramatic effect on the production of courtship USVs in these males. But in contrast, when we look at the total amount of time that the mouse spends engaged in non-vocal courtship behaviors, we do not see any significant effect. And so this is, again, evidence that supports the idea that these cells play a fairly selective role in the control of USVs, as opposed to playing a more generalized role in sexual motivation or something along those lines. And then to the final of the 3 questions that we set out to ask, we wanted to understand whether these PAG-USV neurons were important for gating the vocalizations or alternatively for patterning the timing of individual vocal elements. So the first thing I'll point out is that when we compared the overall duration of a particular laser stimulus that we use to the duration of the resulting vocalization, you can see that they map pretty well onto one another. And I hope you can see that in the spectrograms as well. So if you do a 2-second laser pulse, you tend to get a 2-second long or perhaps a bit shorter bout of vocalization. Another thing that's worth noting is that from these raw examples alone and more detailed quantification that I don't have time to show today, you can see that patterned optogenetic stimulation is not necessary to get patterned USV production. So here in the second example, we're giving a single tonic light pulse, and even within a single tonic light pulse, you get multiple USVs. And so we made more detailed measurements of the respiration. And in fact, what is going on is that you're getting USVs when the blue light coincides with periods of exhalation. So mice are like us and they produce vocalizations as they're exhaling. And furthermore, I'm not showing you today, but we established that the ongoing respiratory rhythm was not altered by optogenetic activation of these neurons. So all of this is consistent with the idea that these cells are gating the production of USVs, but they're not necessarily involved in the fine temporal patterning of individual vocal elements. So that's where we started in the midbrain. I'm going to walk you now through a second story, which is, as I said, it's largely finished. It's in review now, but it's been on bioRxiv for a bit, and we put a more updated version really just this last month. But what we did was we went upstream from these PAG-USV neurons, and we wanted to understand what are the cells that provide input to the vocalization circuit within the midbrain, and how do they shape vocalization? So, of course, an animal is not vocalizing in a vacuum. It has to decide whether or not to vocalize in a manner that is matched to the ongoing social context. And so that's sort of the bigger picture goal of these sorts of experiments, is to understand how that process occurs and what the brain mechanisms for that process are. So to begin with, we started again with these midbrain neurons that we just talked about. And we performed rabies-based monosynaptic tracing from these PAG-USV neurons to identify neurons throughout the brain that provide direct input to these cells. And so we did this tracing actually, in addition to doing the tracing from the PAG-USV neurons, we also did the tracing from nearby GABAergic neurons within the PAG. And without going into the details, we have evidence, and there's a lot of evidence accumulated from other studies that indicates that excitatory projection neurons in the PAG are subject to really strong regulation by local inhibitory neurons. And so we reasoned that by doing tracing from both of these cell types, we could get some insight into which forebrain neurons are really gaining access to the midbrain vocalization circuit. So this slide is a little bit of a data dump, but it's intended to show you that, of course, we see inputs from many different parts of the forebrain. So it's not just 1 or 2 regions. We see inputs to the midbrain vocalization circuit from a really big variety of forebrain regions. So of course, we didn't study all of those. So let's now zoom in to the couple of regions that we did end up studying. So there are 2 regions. The first is the preoptic area of the hypothalamus, which I'm showing you in this top panel here. And then the second region was the amygdala. And we actually saw a fairly restricted pattern of labeling within the amygdala. We saw this in the final row, this nice pocket of labeling within the central amygdala. And then if you continued rostral from there, we saw this kind of continuous strip of labeling And we've given it sort of a strange name. This is intended to represent a central medial boundary zone of the amygdala. And so this strange little pocket of cells, it basically sits in a region that straddles the central and the medial amygdala. It's not really squarely in one or the other, but it sits right in between the two. So there was really this stretch of labeling in the amygdala going from those cells back to the central amygdala. And for the sake of time, I haven't included everything here, but I'll just say that the viral strategies that we use to label these cells that were all AAV based for the experiments that follow, there just must have been some issues with viral tropism. And at the end of the day, our strategies just did not label this pocket of cells within the central amygdala, despite the fact that they're labeled quite robustly using the rabies-based tracing. And so the two regions I'll talk to you about today then are the preoptic area and then this kind of central medial boundary zone of cells, which was really well labeled by our viral strategies. And so backing up, a little bit, why these two regions of all the regions that we saw labeled? The preoptic area is kind of a no-brainer in the sense that it's been known to play an important role in sexual behavior in a variety of species. And more specifically, it's known to play a role in the production of courtship vocalizations in both rodents and songbirds. So it was a pretty obvious reason to look at this region in particular. And of course, the hypothesis is that we think these cells will promote the production of ultrasonic vocalizations. In terms of the amygdala, we are kind of drawn into it just by the fact that not so much is known about how the amygdala contributes to vocalization. So it, of course, is known that the amygdala generally plays a role in the processing of social information, including vocalizations. And there is some work out there, really nice work actually, that does implicate the amygdala in regulating the production of vocalizations, and particularly in regulating production of vocalizations that are produced in response to hearing a vocalization. But in any case, there really is a lot left that we don't know. And so we decided to tackle the amygdala in part just because we didn't know exactly what to expect. So we'll start with the POA. And so I hope you can see here that our hypothesis turned out to be correct, and optogenetic activation of these cells really strongly promoted USV production. So in the strategy that I'm showing you here, we're injecting a retrogradely infecting AAV into the PAG, which is driving Cree expression. And then locally within the POA, we're injecting a Cree-dependent channel rhodopsin virus. And so what we're doing is expressing channel rhodopsin and POA neurons that project to the PAG. So you activate them and you get really robust USV production. That's true for many different trials in this mouse, as well as for many different mice in the data set. And it was also true for both males and females. So we know from a lot of really nice work by their labs that many neurons within the preoptic area express estrogen receptor type 1. And so we wondered next if we could take advantage of this, activate ESR1 positive neurons within the POA and recapitulate our effects on vocalization. So what I'm showing you here is that we first established that the cells we're labeling using our viral strategy do in fact express ESR1. And so with that knowledge in hand, we went ahead and we used an ESR1 Cree mouse to activate neurons, ESR1 positive neurons within the POA. So we get USVs if we activate those cell bodies. If we activate the axon terminals of those neurons within the PAG, we also get vocalizations. So it appears to be ESR1 positive neurons that project to the PAG that are really the key players here. And I'll just say briefly that we did not see USVs elicited by optogenetic activation in a variety of control experiments. So onto the amygdala then. Turns out these neurons are inhibitory as well, although I'm not showing you those data here. And so we're using a very similar strategy to optogenetically activate these neurons. So what we're employing similar strategies as in the previous experiment, but this time looking at amygdala neurons that project to the PAG. And I'll say first what we saw, which isn't shown here at all. We tried activating these cells in mice that were alone in a cage and nothing in particular happened. There's just no overt effect at all on behavior. So we scratched our heads a bit and then reasoned that perhaps these cells, rather than promoting vocalization, actually inhibit vocalization. And it turns out that's what we see. So we see that really robustly across many different mice. This light-locked decrease, transient decrease in the USV rate. And I'll say also that we ran a variety of control experiments. Of course, if you're shining blue light on a mouse's head, you always worry that what you're doing is simply scaring the animal and it has nothing to do with your manipulation of neural activity. And in fact, that's not what we saw. So the black, the green, and the gray all represent different control experiments. And so if you look from pre to the laser period to post, you do see a decline in USV production over time, but that actually represents the natural statistics of USV production. So as time goes on, measuring from the beginning of a vocalization bout, the mouse is more and more likely to stop vocalizing as time goes on. And so you see this big dip in our true optogenetic experiments that you never see in the control groups. So putting these two stories together, I mentioned both of these neurons are inhibitory populations of neurons. So one promotes vocalization, one inhibits vocalization. How does that work? I didn't have time to show you today, but Valerie Michael, a grad student in the lab of Rich Mooney, who I collaborated with for this project, has done a series of really elegant and beautiful slice experiments to show very convincingly that the POA neurons, in fact, predominantly provide synaptic input to GABAergic neurons within the PAG, which in turn provide input to PAG USV neurons. And so it's this disynaptic mode of effectively they're disinhibiting the PAG USV neurons to promote vocalization. In contrast, the amygdala neurons provide direct inhibitory input to the PAG USV neurons. The idea is they're just really directly clamping vocalization via that synaptic input. So in the couple of minutes that remain, I want to leave you with some current directions that my lab is beginning to investigate at Cornell. So first of all, vocalization isn't just binary. So a mouse needs to decide, or an animal in general needs to decide whether or not to vocalize, but also what type of vocalization to produce, right? And so, I'll say again that mice produce USVs in a variety of social contexts, so opposite sex and same sex social interactions. And they, of course, also produce different types of vocalizations, including audible squeaks during, painful, scary, aversive situations. And so one thing that we're beginning to investigate is really how specialized versus how general are these PAG USV neurons that we first characterized. So are they really all-purpose neurons that control USV production across many different social contexts? Or perhaps they're even more general than that, and they actually control the production of many different acoustic categories of vocalization. And we are really just beginning this work, but we have some evidence that indicates, that they're really probably all-purpose cells for USV production, but that a different population of midbrain neurons is important in the production of audible vocalizations. A related question is understanding whether the same population of PAG neurons is important for the control of USVs produced across development. So I mentioned at the beginning that mouse pups produce vocalizations, but the stimuli that trigger vocalizations in a pup versus an adult mouse are very, very different. So in the pup, they're vocalizing primarily when they're isolated and especially when they get cold. And so you'll see this profile of vocalization as follows. Vocalizations increase during periods of isolation, and they actually decrease with social contact. In contrast, in the adult, they don't vocalize much when they are in isolation, and in fact, vocalize quite a bit when they are engaged in social contact. And so what we're really wondering is it the same cells in the PAG across development that are important for USV production? We suspect that it is, but in that case, it must be that the forebrain inputs, or just generally the inputs to these cells, are changing in some dynamic and interesting way across development to allow for the gating of the behavior in response to such different stimuli across development. So that's really the upshot of the second project that we're starting in on. The third thing I'll say is that, of course, we're interested not only in the midbrain, but in inputs to the midbrain. And so to really kind of follow up on the second story I told you about, we're broadly interested in characterizing forebrain inputs to this circuit in the midbrain, trying to understand whether there are any inputs that are really acting as drivers of vocalization, that are playing, a really central and crucial role in the production of vocalization versus others that might be more modulatory in nature. And another question we're really gunning after is to understand whether there are forebrain inputs that are important for the production of vocalizations across multiple different contexts. And so one thing I'll say about that is that in many different social contexts, USVs are produced by mice during periods of social investigation. So one possibility is that there are some sets of forebrain neurons that are particularly important for promoting social investigation, and that these might be key drivers of this midbrain circuit across a variety of social contexts. And the final thing, and I think a really, really important thing, is to leverage these tools we're developing to better understand the communicative functions of USVs. So as many people know, mouse models are used all the time to model human disorders that are characterized by communication deficits, including autism spectrum disorder. But it's been very difficult to understand whether and how vocalizations, or USVs specifically, are important for social behavior and or the social success of mice. And the reason is that they're produced in conjunction with non-vocal social behaviors, and without a way to selectively manipulate the vocalization, it becomes really difficult to know exactly what the vocalization is contributing for the social life of the mouse. So the plan going forward in this arm of research is to leverage these tools we're developing to characterize and to manipulate the activity of midbrain and forebrain neurons that are important for vocalization, and muck with the circuits effectively. So pair these circuit manipulations with both short-term and long-term measurements of social success, including reproductive success, social rank. How do these manipulations affect the ability of a mouse to access resources and to acquire and defend territories? And we have experiments planned both in the lab and in the field. And the field experiments that we're starting to plan out are being done in collaboration with Dr. Mike Sheehan, who's also at Cornell. So I will end there. And I just want to say it's been a really weird time to start a lab in the middle of a pandemic. And I am so incredibly grateful to the students and postdocs who have bravely joined my lab at this juncture in particular. So a huge shout out to Nicole, Pat, and Shin. And also to the team of undergrad RAs that we have here, just helping us a ton with our data analysis. And finally, I'll again give a big shout out to my collaborators at Duke, Dr. Fan Wang and Rich Mooney, with whom I worked as a postdoctoral fellow, and particularly to Valerie Michael, with whom I collaborated really extensively in my postdoctoral work. And I will take any questions.
Thank you very much.
Yeah. Is it okay if I stop sharing the screen?
Yeah, sure.
Okay.
Yep. So I want to start off with a question that I have before we get into the questions that the attendees have been asking, which is, so, looking at this from the human side, there's been some speculation that certain chord preferences in humans are related to some degree to innate things that we might have inherited. For example, major and minor preferences are associated, major and minor chords are associated with different emotional tones and balance. Do you think there's any truth to that evolutionary angle in that in particular? And for example, they relate it to dog, dog tones, such, dogs tones going up versus down, indicating aggression or submission.
Yeah, it's hard to comment on that in the mouse. There actually is some really nice work by Josh Neunenbell's lab. He's at University of Delaware, and he has seen associations between in mice that are engaged in chasing interactions. I actually forget which direction it goes, but the story is something along the lines of the mouse chasing is more likely to do an up sweep, and the mouse chasing or being chased down sweep. I might have it backwards, but there's some difference depending on, are you the aggressor or are you the one being chased? So it's totally possible, but I think in terms of
like
understanding
pathway that's been laid out that would.
No, and it's actually just a really huge open question in terms of mouse vocalizations. Exactly. I'll just say that their vocalizations are enormously complex from an acoustic perspective, and in terms of exactly what that might mean, that's a huge open question that we're really interested in tackling. But yeah, it doesn't exactly get at your question.
Well, it's a hard one. I mean, the thing is, even if you found this correspondence, it's just a flip of a coin whether up means the same thing, right? I mean, it's like a 50, it's not a lot of information there.
Yeah.
Okay, so let me start off with a question here by Steve Shia, who asks, have you tried exploiting your ability to command the mouse to sing to make it sing at inappropriate times to see if it affects reproductive success?
We haven't done that, but we plan to. So I may have framed some of these circuit manipulation experiments like we're going to silence the neurons and see what happens when the mouse can't vocalize. So we'll do that flavor of the experiment. And we're also planning on doing the opposite. So have the mouse vocalize at inappropriate times. Does that negatively impact reproductive success? We're also right now trying to look at the relationship between vocalization during same-sex interactions and how that might relate to social rank. And kind of a similar set of experiments, if you make the mouse vocalize, not necessarily even at inappropriate times, but just more than it otherwise would, does that mouse become more dominant? Is there any directionality in that sense? So we are planning to do that general type of experiment, yeah.
Okay, next question here. I think male mice, not females, mainly release USVs during the copulation. Your study showed MPOA stimulation of females induced USVs. Do USVs by MPOA stimulation between males and females show the same pattern? In other words, can we say that female mice also have long love song like males? Is MPOA in female active during female-female interaction rather than male-female interaction? So there's a lot of things to unpack there.
I think I'm getting the gist of the question though. Yeah, I mean, so a couple of things to note are, I mean, I don't want to say that we like directly compared the acoustic features of like the male versus the female POA elicited USVs, because I don't think we actually did that. One thing I will note, though, is that there's actually a study by another group. It came out a bit before. Well, ours hasn't come out yet at all, but it came out sometime in 2019 that showed actually the POA isn't necessary for these vocalizations. So even in a male mouse, if you ablate the POA, they still produce USVs. And so it does highlight, some of the shortcomings of optogenetic experiments. Just because something can do something doesn't mean it does necessarily. So we think, I mean, I suspect that in the case of the POA and how it contributes to vocalization, it's probably, it might be promoting them or playing a more modulatory role in shaping the acoustic features of the vocalizations to be more characteristic of courtship vocalizations, for example. With regard to its role during natural behavior in female vocalizations, they do produce vocalizations during courtship, a small percentage of them, but particularly when they're in estrus, they do. So it's possible that it's active then, although we don't know. And it's also completely possible that it's active during same-sex interactions in females, but same answer, we don't know. But in any case, I think given that a total lesion of the area or ablation of those cells doesn't impact USV production in a coarse sense, it's probably not. It's more like a modulator, not a driver of the behavior.
All right, so the next question then is from Billy Lau. He says, wonderful talk. I was wondering if you could elaborate a bit more on the pup vocalizations and social interaction part, where you mentioned that the USVs decrease in pups in social contact. What context is this? Pup retrieval or just one-to-one interactions between adult and pup?
Yeah, I guess what I'm referring to there is like a little more specific than what I really said, which is that, for example, if a pup has been isolated from the nest, it gets cold exactly during pup retrieval, that social contact actually reduces the vocalizations, whereas you see kind of the opposite pattern in the adult where they, alone, I mean, obviously, even if they're alone and cold, they're not going to be producing USVs, but then upon social contact, that's really when the USVs start. So what we're really getting at there is like what's going on from a neural circuit level that allows, thermal cues essentially to drive USV production in a pup versus social cues in the adult. And we're also conducting some, just some more basic behavioral experiments to get some sense of exactly how that sensitivity to social cues of different sorts arises over development and how variable it is across different animals, et cetera.
Is that a sort of generic distress cue in the pups?
Yeah, it's funny, right? Because the pups, it is. It's interesting though, because when the pups, especially when they're really young, they can't even hear themselves doing it. So it's not like some people liken it to the cry of an infant. I'm not really sure if that's like quite the best analogy, but despite the fact that the pup can't hear itself doing it, it's just like really quite involuntary behavior. It's related to laryngeal breaking, as far as I have as far as that's my opinion. I don't know that it's really anxiety or distress per se, beyond what might be expected in terms of physiological distress from decreasing body temperature, at least in these really young animals. But very clearly the dam is using this as a cue to find and retrieve the pup.
Yeah.
So it's still a communication in that sense. Absolutely.
Okay. We actually have a question from YouTube being ported over by Ryan, our backend host, who asks, so Cesar Vargas asks on YouTube, in terms of context-based vocalizations or USVs produced during investigation, do you think the inputs would be patterning the PAG circuit directly or through parallel pathways modifying output?
Yeah, I'm assuming like by patterning, he's referring to like the acoustic, the spectral features of vocalizations. So. Yeah, I mean, it's actually a really tricky question because it's like you say, okay, forebrain neurons are important for shaping how, differences in vocalization and acoustic features across context. But then it's weird, right? Because if all the midbrain is doing is really acting as a gate, how does that signal get through to actually drive these changes in acoustic features? So I think there are a number of possibilities. I think there could possibly be something more interesting going on in the midbrain than people think, and we're going to at least explore that possibility. Although that's not really like my pet hypothesis. I also think it's possible that there could be, and in some cases there are known to be descending forebrain inputs, not only to the PAG but also to the premotor centers that lie downstream of the PAG. And then a third thing I'll throw out there is that to some extent, the acoustic features of the vocalizations are going to be hugely dependent on the respiratory pattern. And so you can imagine, that's going to be shaped. Well, I guess my point is like, if I am saying something to you and I'm walking versus like sprinting, it's going to sound quite different. And that's kind of really an indirect result of the fact that I'm just engaged in different non-vocal behaviors while I'm saying that thing. So I think some of that could be happening as well with the mouse, where you get these indirect effects on the acoustic output that, aren't necessarily really related to the vocalization circuit in such a specific way.
Sort of in effect using the body to do it, right? So I mean, yeah. Would it be fair to say that the signal that produces these USVs could be as simple as, tightening up the vocal folds so that whatever respiration is going on is now capable of causing that output, or is there a more complex motor command that's necessary to evoke them?
Yeah, I think it's a little bit more complex than that, and there has been a lot of really nice work done looking at kind of the bioacoustics of like what what's actually changing at the level of respiration and at the level of the tension of different laryngeal muscles that characterizes USVs versus audible vocalizations versus just like resting respiration. So I do think it is, I mean, it is definitely a little more complex than that. But I think, from what we've seen with our manipulations of PAG, like it does seem like to some extent these descending vocal commands are just, like they're not driving the patterning of the vocalization, certainly. So there's, like your respiratory CPGs that lie downstream, and that vocal command can really just sit on top of that. And the respiratory CPG just does its thing, and then you get these vocalizations just kind of at the correct windows of the respiratory cycle. So yeah, it's something along those lines.
I always find it fascinating to think about how the brain